Hello, I'm Ryan Stewart. I'm an associate professor at the School of Plant and Environmental Sciences at Virginia Tech, and my presentation today is on quantifying dynamic soil properties for soil health in Virginia. And just to note, this research was supported by the NRCS through award number that you can see on the screen. So this is a new project we just got going in October of 2021. So these are some initial results and more of the project context that I want to share with you today. So this is a paper that came out last year, 2021, in the journal Soil Biology and Biochemistry, and it's titled The Soil Health Metaphor, Illuminating or Illusory. And we've been discussing this paper, within our own state, within our own people working in soil health, because at least in my opinion, there's a lot of interesting ideas that come out of this. In terms of data and framing, one of the figures in the paper talked about and compiled the number of documents per year that mention soil health in the title, abstract, or keywords. And we can see this exponential growth in terms of the number of citations per year that are talking about soil health. This paper, in terms of its scope and its ideas, it's really making the argument that soil health is a metaphor, right? This is not something that's tangible, but it's something that we can use to describe how soil responds to management and really putting soils in the context of their ecosystem, of the land, and of the use that we want for that soil. And I think that's a valuable thing to keep in mind. The paper also talks about soil health being this more holistic, larger scale term that encompasses both, the classic soil fertility, soil quality, and gets into human well-being, aesthetics, habitat, climate buffering, all of these larger scale ecosystem services and results that we want soils to be able to provide. A caution from that from that paper is to be specific about what we mean by soil health, right? We need to define it. And that ties in well with some of our efforts here and some of my efforts working with the Virginia State NRCS office, who in May of 2020 came out with their own strategic plan for soil health. And one of the takeaways from that document was they are making the recommendation or the suggestion to define what is soil health as really the first step of any soil health project. So here today, I'm going to work towards our big question that's motivating a lot of the work, a lot of the partnerships that are happening now between Virginia Tech and Virginia's state NRCS. And the core question is, if we apply 4 soil health principles, and these soil health principles have been around for a while, here in Virginia, there's an effort to really take these ideas and bring them out to the mainstream. We have this For the Soil campaign that really emphasizes these 4 soil health principles: minimize soil disturbance, maximize living roots, energize with diversity, keep soil covered. But our question in this realm is, if we're applying these principles to our management practices, do soils really change? And if so, how much do they change? And that's a natural tie-in and segue to this DSP project, where we're trying to quantify these dynamic soil properties in terms of soil health practices within Virginia. And our goals here are to assess dynamic soil properties on different sites under contrasting management or differences in management, different levels or degrees of those 4 soil health management principles. And we're also, like any of these DSP projects, trying to ensure that there's consistency in the inherent soil properties across these sites. So like I mentioned, our DSP project is very new. We just got it going last summer and fall. And so right now we're kind of on this 2-pronged attack, trying to look at DSPs across the state of Virginia. And so this is a pretty large undertaking. There's a lot of key personnel, including myself working at Virginia Tech, and Lydia Fitzgerald, who is in a pretty special role, a pretty neat role, where she's a Virginia Tech employee, but she's working in the state NRCS office, actually in a regional office, but interfacing with that state NRCS office. On the NRCS side, Chris Lawrence is the main contact point, the main motivator for a lot of these efforts. And right now we're focusing our efforts on 2 parts of Virginia. The Shenandoah Valley is, in the northwestern part of the state, characterized by a lot of some dairy operations, but a lot of grazing of sheep and cattle. And so kind of leading those efforts is Alexandra Schmidt, who works in the Rockingham office. She's a soil scientist and really the motivator for a lot of this project that I'm going to talk about today. We also have a parallel project taking place in the coastal plain. And here in Virginia, the coastal plain is much of our row cropping. We have tobacco, cotton, corn, wheat, soybean, all the usual suspects. And so that effort's being led by Carl Peacock, who also is a soil scientist working with NRCS. So today, for this short presentation, I'm going to focus on the Shenandoah Valley, this Valley Pasture DSP project, and the pilot study that we conducted last summer of 2021. And my counterparts at the Virginia NRCS office tell me that this is the biggest undertaking they've ever, attempted in terms of soil health assessment. And as I'll go through today and explain, we learned a lot of lessons along the way, but this really involved a lot of human resources, a lot of capital, a lot of time, people from different MLRA offices coming down, different different field offices, all coming together, coordinating to collect these data. Like, many DSP projects, our typical site layout, we had a main paidon, and then we had 2 satellite paidons, approximately 10 meters spacing between them. And in this pilot project, we wanted to look at differences in dynamic soil properties based on grazing practices. So we tried to categorize our pastures in one of 2 categories. On the left is rotational grazing, where the farmer's doing some kind of rotation, getting animals in and out of fields, trying to minimize traffic, things like that. And so we found 5 sites that met this description with 3 paidons each. We had 15 profiles that we worked with. And we were trying to contrast that with more of a conventional grazing where the animals are going to be in one field kind of more consistently. We would assume more traffic, maybe more soil compaction. And our hypothesis, and so we had 4 sites, 3 paidons, 12 profiles. Our hypothesis going in was that the rotational grazing sites were going to have healthier soils. We're going to have higher values for a lot of these dynamic soil properties that I'll talk about in a second. Here's just the spatial distribution of these sites. You can see pretty close together. One site, one of the rotational sites was a little bit farther away, but all these sites fell on a benchmark soil series, the Frederick Silt Loam, which is a fine, mixed, semi-active, mesic, typic paleoldalt. As we found out, and as we continue to find out, the site history is really important, particularly working in these pasture systems. And so here's just, one of the things that we have been working to piece together is what is the previous management? Have these sites been managed previously in cropland? And so all these sites, with the exception of one, all these sites have been in pasture for at least 10 years. And only one of the sites, there was a known history of cropland before that conversion to pasture. So we're starting out, fairly uniform, at least for the known history of these sites. And the other factor I think that we didn't appreciate at the time, but has become more important as we kind of go and do our homework, is that all of these sites were involved in NRCS programs to some extent, right? So one or more NRCS conservation program has been implemented in there. So, we don't have any sites that are really terribly managed, poorly managed. And again, I think that's going to be something that has an effect on the results that I'm going to share today. We also looked at what's known as the pasture conditioning score, which is a way to describe attributes of a pasture system. And so we can see the PCS score here. We had a range of 26 on the low end to 45 on the high end. And this likely response to new management comes from the NRCS document. So on the low end, we would expect to see moderate improvement in the pasture quality if we improve our management. And on the high end, there's really very little benefit to changing management because things are pretty good. And just a note for on PCS, you can easily see values around 15 on the low end. And so we didn't really get all the way down to a really degraded pasture system, but 45 is pretty high. The maximum score is 50. So, we definitely did capture what would be a pretty well-managed pasture system in here. On all these sites, we analyzed a lot of DSPs, including some chemical ones. I'm going to focus today on total carbon in my presentation. Physical properties, I'm going to focus on bulk density measurements and then the single ring infiltration, where we infiltrated 2 inches of water and timed each of those infiltration. And then we've been looking at a lot of these biological indicators. There's a lot of interest from, again, NRCS here in Virginia on these biological indicators: active carbon, soil protein, the CO2 burst test, mineralizable carbon, and the total carbon as well. And so jumping into results, when we looked at total carbon and did a conversion to organic matter, we saw that there was a slightly higher numeric value in the rotational plots in terms of carbon and organic matter, but not a significant difference here. And all of these organic matter percentages are pretty high for Virginia, 5 to 6%. When we looked at some regression against that pasture condition score, the PCS, we saw that there was potentially a relationship, particularly in the surface sample, the 0 to 2 inch depth, but kind of contrary to what I would have expected, we have a negative relationship. So the higher the PCS, or the better managed the pasture, the lower the carbon level. And I'll just note that there are one site in particular had really high carbon contents on the order of 6 to nearly 8%, which is much higher than we expected, and we need to do some more work to verify if those values are real and what it means and what's going on in that pasture in particular to give such high carbon levels. Bulk density, similar story, that the rotational sites had a numerically lower bulk density than the continuous graze sites, but again, those differences weren't significant. And here we saw what is a potential relationship with that PCS score. But again, contrary to what I would have expected, in the sense that the higher the PCS score, the better managed the pasture, we actually saw higher bulk densities. And going into this, I would have thought we would see the opposite, that the better managed pastures are going to be lower in bulk density. And so I think there's something going on here between bulk density and the amount of carbon in the soil, where we do see this inverse relationship. This is something that's been seen in other studies. You get more carbon in the soil, you're going to decrease your bulk density. And so something's going on in the sense that the higher PCS pastures have lower carbon. That's translating to higher bulk density. And so, we don't fully understand at this time why that is, but it's something that we're trying to dig into and better understand going forward. In terms of our infiltration behaviors, again, we did just that single ring infiltration, put in 2 inches of water, timed the first inch, and then again the second inch. And so when we look at these two categories, continuous grazing versus rotational, we see that there's higher rates in the rotational pastures than the continuous. And in general, we went out there in the middle of summer, the ground was very dry. We had very fast infiltration rates for that first inch, and then things slowed down a lot for that second inch, pretty much at all the sites. And so that second infiltration rate is probably more indicative of things like the hydraulic conductivity of the soil or the permeability of the soil. And so when we looked at that infiltration rate for the second inch versus the PCS, we again saw a relationship that may be significant, maybe not. We haven't tested it yet, but again, is contrary to what I would have expected. The lower PCS score sites, or the less well-managed sites, had a higher infiltration rate compared to the higher PCS scores. So one of the takeaways for us is that PCS may not be the best kind of explanatory variable here for trying to understand and interpret differences between these pastures. So that's something that we're going to try to get a better handle on going forward. Quickly on biological properties, and so we just picked here 4 sites, trying to get a range of our lowest PCS score was site 3, our highest was site 2. And when we look at these different indicators and the mean values, we see that the active carbon or epoxy, there's some variation, and it seems to somewhat follow that the rotational sites had a higher active carbon than the continuous, but not a lot of dynamic range there. The values are pretty clustered together. In terms of the ACE protein, even more tightly clustered. One of the sites was 15.7, but everything else was kind of right in that 13 to 14 range. Our mineralizable carbon, we see a difference of almost a factor of 2 between, site 4 and site 2. And then our CO2 burst test, that 3-day incubation and rewetting of air dried soils, we see almost a factor of 3 difference between site 4 and site 2. So that's an indication that this mineralizable carbon, the CO2 burst test, may have more variation within these pasture systems. And that's something we're going to try to figure out more going forward. A quick note and a caveat, something else we found, we took our soil samples, we did some subsamples of those samples, prepared them, and sent half off to the Cornell Soil Health Test Lab, and then did our own in-house measurements of that ACE protein. And we saw that there wasn't a great correlation between the two. Some of the values fell right on that 1-to-1 line, but a lot of them seemed to have higher values in terms of the Cornell measurements compared to what we were measuring in-house. So that tells us we may need to fine tune and tweak our own procedures, but there also just may be some inherent lab to lab variability in these properties that we need to get a better handle of. And the last thing I'll talk about, I'm a little bit over time, is our 3-day CO2 burst test results. Again, I've plotted the CO2 burst against that pasture conditioning score. And so when we take mean values from our sites, from our 9 sites, there's almost no relationship, R squared of 0.8. But if we break this down by sampling depth, and so on the left, I have our surface 0 to 2 inch samples, and then on the right, I have our 2 to 4 inch samples. All of a sudden, we start seeing, particularly in the continuous grazing sites, that there may be a correlation, that the higher the PCS, the better the CO2 burst test, the higher the CO2 burst test values are. And when we talk about that slightly different, or slightly deeper sample, the 2 to 4 inch, we see both in the continuous and the rotational, what appears to be significant correlation between the two. So again, this tells us that maybe we need to be more strategic with what we're using to explain differences in the sites or how we're characterizing the sites. And we also maybe need to be strategic with what depth we're working with and where we're taking our samples. And so to wrap up, this was, like I said, a really big undertaking, particularly from the NRCS side of things, who really led the way in terms of getting people out there, coordinating, doing all the heavy lifting. And so we've learned a lot of lessons, some the hard way, that's going to really inform our efforts going forward and allow us to hopefully be successful with both this pasture project, as well as we start getting more in depth into our coastal plain sites, where I expect the contrasts in land management are going to be much more apparent, and hopefully we'll see even more variation in our dynamic soil properties. So with that, I'll wrap up. Thank you for your time and have a great day.